A scale plate on a piano keyboard and a gene on a chromosome are both a series of simple linear elements, keys or nucleotide pairs, that produce information. A wrong note or an altered nucleotide pair calls attention to the structure of the musical scale or the gene. So, what is genetic mutation? Well, a genetic mutation is a change in a sequence of your deoxyribonucleic acid. Your deoxyribonucleic acid sequence gives your cells the information they need to perform their functions. If part of your deoxyribonucleic acid sequence is in the wrong place, isn't complete or is damaged, you might experience symptoms of a genetic condition. Human chromosome 3 consists of approximately 220 million base pairs and carries about 1,600 genes. Somewhere on the long arm of the chromosome resides the gene for rhodopsin. It is a light-sensitive protein, active in the rod cells of our retinas. The rhodopsin gene determines perception of low-intensity light. People who carry the normal wild-type allele of the gene see well in a dimly lit room and on the road at night. One simple change, which is a mutation in the rhodopsin gene, however, diminishes light perception just enough to lead to night blindness. Other alterations in the gene cause the destruction of rod cells, resulting in total blindness. Medical researchers have so far identified more than 30 mutations in the rhodopsin gene that affect vision in different ways. The case of the rhodopsin gene illustrates some very basic questions. Which of the 220 million base pairs on chromosome 3 make up the rhodopsin gene? How are the base pairs that constitute this gene arranged along the chromosome? How can a single gene sustain so many mutations that lead to such divergent phenotypic effects? So, mutations may be classified by how they change deoxyribonucleic acid. So, let's discuss types of genetic mutations. A substitution occurs when a base at a certain position in one strand of the deoxyribonucleic acid molecule is replaced by one of the other three bases after deoxyribonucleic acid replication. A new base pair will appear in the daughter double helix. Let's understand it. Suppose this is starting sequence. Observe at position 9. Guanine in upper stand and cytosine in lower stand. Substitutions can be subdivided into transition, in which one purine replaces the other purine, or one pyrimidine replaces the other pyrimidine. As you can see at position 9 of starting sequence, guanine and cytosine are replaced by adenine in upper stand and thymine in lower stand. And other subdivision of substitutions is transversion, in which a purine changes to a pyrimidine, or pyrimidine changes to purine. At position 9 of starting sequence, guanine and cytosine are replaced by cytosine in upper stand and guanine in lower stand, or at position 9 of starting sequence, guanine and cytosine are replaced by thymine in upper stand and adenine in lower stand. Other types of mutations rearrange deoxyribonucleic acid sequences, rather than just change the identity of a base pair. A deletion occurs when a block of one or more nucleotide pairs is lost from a deoxyribonucleic acid molecule. Let's understand it. This is the starting sequence. Observe position 5, 6, and 7. From the starting sequence, these three nucleotide pairs are deleted or removed. So, this is deletion. And now the new sequence is different from starting sequence. Due to deletion of 3 pairs of nucleotide, last is an insertion. Insertion is just the reverse of deletion. It is the addition of one or more nucleotide pairs. This is starting sequence. To the starting sequence, if 2 pairs of nucleotides are added, at position 5 and 6, the insertion occurs, and there will be new mutated sequence. Deletions and insertions can be as small as a single base pair, or as large as megabases. That is, millions of base pairs. Large deletions and insertions are only some of the complex mutations that can reorganize genomes by changing either the order of genes along a chromosome, the number of genes in the genome, or even the number of chromosomes in an organism. Now, how natural processes can change the information stored in deoxyribonucleic acid? Well, in depurination from deoxyribonucleic acid strand, the hydrolysis of adenine or guanine bases leaves a deoxyribonucleic acid strand with an unspecified base. This is deoxyribonucleic acid strand, with guanine attached with it. Now hydrolysis will occur. Deoxyribonucleic acid strand will be left, and guanine is released. That the hydrolysis of a purine base, adenine or guanine, from the deoxyribose phosphate backbone occurs 1,000 times, an hour in every human cell. This kind of deoxyribonucleic acid alteration is called depurination, because the resulting apurinic site cannot specify a complementary base. The deoxyribonucleic acid replication process introduces a random base opposite the apurinic site, causing a mutation in the newly synthesized complementary strand 3/4 of the time. Second is the deamination. In deamination, the removal of an amino group from carbon initiates a process that causes a transition after deoxyribonucleic acid replication. For example, cytosine has amino group. From it, amino group will be removed in deamination. So, deamination can change cytosine to uracil. Uracil is found in ribonucleic acid, but not in deoxyribonucleic acid, because uracil pairs with adenine rather than guanine. Deamination, followed by replication, may alter a cytosine and guanine base pair to a thymine and adenine pair in future generations of deoxyribonucleic acid molecules. Moving forward, X-rays break the sugar phosphate backbone and thereby split a deoxyribonucleic acid molecule into smaller pieces, which may be ligated back together improperly. This is starting sequence. When X-ray falls on it, 3 base pairs will be removed, and a new mutated sequence will be formed. Ultraviolet or UV radiation causes adjacent thymine to form dimers, which can disrupt the readout of genetic information. This is sugar phosphate backbone, with thymine attached with it. Ultraviolet or UV radiation causes adjacent thymine to form dimers. Thymine dimer is formed as a covalently bonded complex of two adjacent thymine on a single strand of deoxyribonucleic acid. Irradiation causes the formation of free radicals, such as oxygen molecules with an unpaired electron, called as oxidation, that can alter individual bases. So, irradiation causes guanine to form free radicals, such as oxygen molecules with an unpaired electron, if not repaired before deoxyribonucleic acid replication. All of these changes alter the information content of the deoxyribonucleic acid molecule permanently, moving to deoxyribonucleic acid polymerase's proofreading function. If deoxyribonucleic acid polymerase mistakenly adds an incorrect nucleotide at the 3' end of the strand it is synthesizing, the enzyme's 3' to 5' exonuclease activity removes this nucleotide. 3' to 5' exonuclease activity cuts here. Now, in next step, wrong base is excised. In last step, correct base is added by deoxyribonucleic acid polymerase. So, this process gives the enzyme a second chance to add the correct nucleotide. Remember that. An alteration that occurs in deoxyribonucleic acid is heritable only if deoxyribonucleic acid repair fails. To reverse the change before the next round of deoxyribonucleic acid replication, the bacterial resistance is effect of genetic mutation in bacteria. So, plating verifies that bacterial resistance is the result of pre-existing mutations. This is called as the replica plating technique. Master plate with penicillin resistant and penicillin sensitive bacteria, and no penicillin in medium. Invert master plate, pressing against velvet surface, leaves an imprint of colonies. Save plate. Invert second plate, called as replica plate, pressing against velvet surface, picks up colony imprint. Replica plate has penicillin in medium. Then incubate plate. Only penicillin-resistant colonies grow. Compare with position of colonies on original plate. So, conclusion is that pressing a master plate onto a velvet surface transfers some cells from each bacterial colony onto the velvet. Pressing a replica plate onto the velvet then transfers some cells from each colony onto the replica plate. Investigators track which colonies on the master plate can grow on the replica plate. Here, only penicillin-resistant ones. Now, let's confirm that. Mutations occur prior to penicillin exposure. Master plate with 107 colonies of penicillin-sensitive bacteria. Master plate has no penicillin in medium. Invert master plate. Pressing against velvet surface leaves an imprint of colonies. Save plate. Make 3 replica plates. Incubate to allow penicillin-resistant colonies to grow. Penicillin-resistant colonies grow in the same position on all 3 plates. Remember that. All 3 replica plates have penicillin in medium. So, Conclusion is that colonies on a master plate without penicillin are transferred sequentially to 3 replica plates with penicillin. Resistant colonies grow in the same positions on all 3 replicas, showing that some colonies on the master plate had multiple resistant cells even before exposure to the antibiotic. So, this was all. Stay tuned to sqadia.com for more precise and knowledge-stuffed videos.
Have you ever heard of video books? sqadia.com is delighted to present video books, medical books that have been tailored into a series of videos. sqadia.com videos are presented in the same order as the book's chapters. Explore now and make your learning easy.